Hi, everyone. I'm Lisa Reimer. I'm co-editor-in-chief of the journal Medical and Veterinary Entomology. And on behalf of the Royal Entomological Society, we're pleased to welcome you to our webinar on sustainable vector biology research systems in Africa. This webinar accompanies a special issue that we've just published this week under the same name, and this can be available on the Medical and Veterinary Entomology website, or you can follow the link in the chat. So just a few housekeeping items before we begin. This event will be recorded. If you have any technical issues, please leave the call and rejoin. Cameras and microphones will be off for all participants except the speaker. And we'll be tweeting during the event using the hashtag #MVEVectorBio. So please use this hashtag from your Twitter accounts as well. So just to give you an overview of what to expect for the next hour and a half. So following an introduction from our guest editor, Professor Moses Bakri, we'll hear about some of the research conducted under the PVEC Capacity Strengthening Consortium. So PVEC stands for the Partnership to Increase the Impact of Vector Control, and we'll hear from 6 of the research fellows from this scheme. We'll then hear from Dr. Justin Pufford, who will reflect on some of the lessons learned during the fellowship scheme. And finally, Professor Hilary Ranson will be chairing a Q&A with all of the speakers. So please add your questions into the chat, and we'll address these at the end. So with that, I would like to invite Professor Moses Bakri to start us off.
Well, thank you very much, Lisa. It has been a wonderful experience working with PVEC and the Medical and Veterinary Entomology team. And it's been a pleasure going through and learning about all the work that's been going on in Africa when this program was being implemented. So what I would like to talk about today is really share my experience, and just in the context of what the importance of medical entomology is really, and the role of local scientists in trying to ensure that our research is translated into policy and it's of great value to the national programs. So, but first of all, what I wanted to do was to just remind everybody about through the historical context, how important medical entomology has been for the last 100 years. Next slide, please. So very few people are probably aware that the first Nobel Prize awarded for malaria to Sir Ronald Ross in 1902. That was the second Nobel Prize actually for physiology of medicine, and the first British Nobel Prize. That was actually for his work on vector work, on establishing the role of mosquitoes in the field in the transmission of malaria. I mean, there's all this thing about the Italians were involved in it before because they showed in the lab that anopheles were really responsible, but Sir Ronald Ross was the first person to do that. And I think it's been, I was lucky to be born in Sierra Leone where all this started. So I did a review on that many years ago in order to establish that. And it also ties in the history of Liverpool School. And everybody knows about Liverpool celebrating 125 years of its existence next year. But everything started with the Liverpool team going to Sierra Leone to do some work, medical entomology work in the field. And that started in 1899. And so it's not only around working with malaria vectors, but other vectors as well, vectors of onchocerciasis. So the photograph you see on the right is that of Donald Blacklock working in a lab in Sierra Leone in the early 1920s, and was the first person to demonstrate that Onchocerca volvulus was transmitted by black flies. Next slide, please. So, and so I've started about all this work in medical entomology starting in Sierra Leone in 1899, but I mean, no local scientist was involved in medical entomology work, especially with mosquitoes, until about 1989, when I enrolled for my PhD in the Liverpool School of Tropical Medicine and started working on vector biology and malaria transmission in Sierra Leone for my PhD, which was awarded in 1992. So, I mean, that just shows how limited capacity was. So even though there was all this happening in Sierra Leone with all these brilliant people coming from Liverpool School, and establishing good work, but we didn't have local scientists involved until almost 100 years later. So that just shows the paucity and why it is and how it is important now that we are having all this other research happening. Next slide, please. And we have to understand, I mean, for most of us who were studying malaria entomology or malaria epidemiology in the 1980s and 1990s, the GACI project was very, very important. And it was a research on epidemiology and control of malaria in West Africa. And that was started because earlier on, with the global malaria program activities happening mainly in the Americas around Brazil after the discovery of DDT, we had very little capacity for our field work in malaria epidemiology in Africa. So this project was developed really to establish that, so that then you can have also the malaria eradication objective established to control malaria in Africa. The objective was that of the GACI project was actually to use indoor residual spraying and mass drug administration chemotherapy to control malaria. And why was this vector? But in the end, the outcome that was expected of the project was not what they got. I mean, they didn't get the impact that the expected outcome, the sort of significant impact we wanted on the prevalence and intensity of malaria transmission. And the reason for that was because they didn't know that in that area you had Anopheles arabiensis was a lot, and it was exophilic. So even though they were doing this indoor residual spraying, the vectors were feeding outside and resting outside, or even if they fed on people inside the houses, they did not rest inside the houses, they were resting outside. So it actually then demonstrated the value of vector control and vector ecology in these big programs and also in national programs. So over the years that we know now that with the Global Fund and emphasis on all the outcomes, not on entomological outcomes, like here in the GACI project, it was the entomological inoculation rates, the biting rates, but when the Global Fund came, it was like how many kids are sleeping on a bed net, so things really moved. Next slide, please. Now, so this is actually my last slide, and I use it to tie in the value of the PVAC project and how everything has evolved over the years. So we had a lot of expertise going into Africa and in Sierra Leone. So we know about the Liverpool School going there, but also I started my entomology training working for the MRC, just like the MRC in the Gambia. And so there was all this training going on. But I mean, the MRC left Sierra Leone and there was no capacity. So we now have this situation where, I mean, the way this funding has been supporting this research is such that once the funding disappears, I mean, there's very little capacity left. So there's got to be a way of transforming global health through equity-driven funding. And this particular thing, so we should have a situation where people living in Africa must be involved in setting the priorities for global health research policies and programs that affect their daily lives. So I think with the implementation of PVEC and the outcome that will be seen today with so many African scientists working in Africa and bringing this forward, this allows us to focus on the next step, which is how we can now ensure that when funding for programs like PVAC is withdrawn, there has to be a way for African systems to ensure that this happens and the research is sustained. So I hope that we'll be able to learn this from all the other reports we get. Thank you very much.
Thank you, Moses. Tito, are you ready to start sharing your slides?
Yeah, it's done. Yes.
Go ahead.
Okay, thank you very much. Thank you also very much to give me the opportunity to present this work that we did in the framework of PVEC. And the work we are going to present to you is entitled Environmental Mutations in the Campo Focus Challenge the Elimination of Sleeping Sickness Transmission in Cameroon. And to start this work with a small background introduction, the sleeping sickness is this debilitating disease that is caused by the presence in the blood of protozoan parasites of the genus Trypanosoma. And mainly Trypanosoma brucei gambiense and Trypanosoma brucei rhodesiense are two main species that attack humans in Africa. And these parasites are...
Sorry, Tito, to interrupt you. Your slides aren't advancing.
Sorry, Lisa?
Your slides aren't advancing. So
oh. What's
it may be on a split screen.
Sorry, let
Yes.
me try to fix that. Oh, is it fine now? Sorry.
Yes, carry on. Thank you.
Okay. Okay, so I was saying that these parasites are transmitted to human by the tsetse fly of the species Glossina, which are very dangerous flies in that males and females are blood feeding and are both able to transmit the parasite. And also one important aspect in the biology of these trypanosomes is that they can be harbored by many wild and domestic animals as reservoirs, and the flies can take this from the animals and bring back to human. In Africa, it's known that around 250 old and remaining foci exist. And in Cameroon, the disease is mainly present now in one focus, that is the Campo. That is a focus that is shared with Equatorial Guinea, and the border is just the River Thames. So it's actually the main focus in Cameroon and Equatorial Guinea. And the strategy of WHO of screening, diagnosis, and treatment of cases has helped to bring the number of new cases from 500,000 each year in the years 2005 to less than 1,000 today. And this has been very encouraging and pushed the WHO to include this disease or to consider the complete elimination of the transmission of the disease by the year 2030. And you know that as in many parasitic diseases or vector-borne diseases, when we reach at this level, the prevalence is very low, and we need to increase the effort of screening to actually take out and treat the remaining cases, which is very, very difficult to do. And in the areas like Campo, there are many development projects like the creation of big plantation, the roads, asphalting, and also the coming bridge that will cross the Thames River and link Cameroon to Equatorial Guinea, that are bringing many people in Campo and who are carrying out activity that link them or that bring them close to the tsetse vectors. So, as shown already in many other countries, we think that an addition of anti-vector campaign, that's the control of the tsetse fly, may help to tackle this disease. So the aim of this work was to contribute to the elimination of the sleeping sickness in Cameroon. And generally, in this particular work that we are presenting to you, we started by generating the baseline entomological data that will help to guide the vector control. So the work was done in Campo, as already said, at the border with Equatorial Guinea. This is a forest context with around 5,000 inhabitants who actually do the farming, fishing, and hunting that bring them in close contact of the forest flies that transmit the disease. And Glossina palpalis is the most abundant species of tsetse flies there, and unfortunately the one that is responsible of the transmission of the human disease. It is known that in Campo, around 8 new cases occurred from the years 2010 to 2018, and this incidence has increased to 21 cases that were identified in 2019. And we think maybe we understood why, and we will show that to you in the coming slides. So for this work, we performed the evaluation of the tsetse fly densities in Campo by capturing them in the whole territory using these pyramidal blue-black traps that were settled in species that are favorable for tsetse development. And for the tsetse cohort, we counted them, and then we just make a map showing their densities. And then from that, a DNA was extracted and the trypanosomes were detected by PCR-based method. So the principal or the main result obtained there are these two maps that you can see. We did one set of catches in December 2018 and the other one in July 2019. And it is obvious that the tsetse densities are very, very high, up to 100 flies caught per day at a single point where humans are carrying their activities, as you can see in these maps. And also, we detected that around 20% of these tsetse flies were infected by human and animal trypanosomes. And most importantly, Trypanosoma brucei was detected in 4 flies, 1 from December and 3 from July 2019. And remember, in July 2019, the number of cases exploded. And we tried to find an explanation of this, and we understood that in January 2019, a new palm tree plantation was set up, and more than 1,000 people came in Campo and stayed here in Vas, and then somewhere here in Ipono, and were working near a river to produce the young palm trees. And actually, in this point, we identified many of these workers who were actually new people in Campo who were infected in 2019. And also, we caught their flies that were infected with the human parasite. So to conclude this small piece of work, we found actually that there are high tsetse fly densities in Campo that actually can threaten the elimination efforts. And this is certainly linked to the mutations in the area of Campo where there are new people who are unaware of the presence of sleeping sickness who come and settle themselves to work in this forest environment that is close, or that is bring them close together with the tsetse fly that infect these people. And as there are many development projects that are being done in the area, we call the need of vector control in this area. And as this work was done in the framework of the vector control that we plan in the area, these were baseline data that we obtained there. And this baseline data actually guide us to go there with the tiny targets. These are insecticide impregnated traps that are used to control the populations. We call a meeting with authorities to explain what was the use of those traps. As you can see here, we're working close together with local populations, and everyone is now involved or was involved in the 2 years vector control that we started with humans that we were having. As you can see, this military that is on the Entem River at the border with Equatorial Guinea. So everyone there is now actually involved. And in January 2020, we installed the first batch of 2,000 tiny targets that were replaced 3 times for 2 years. So now the project is ended, and we're now seeking for more funds to extend this in the World Campo, and the result of this part of work are to be published in the coming days. So we actually thank all the partners who were involved in this work, and we hope that the future is still best to come. Thank you very much for your attention.
Thanks very much. So we'll move swiftly on to Dr. Koala-san. Tito, you can stop sharing your slide and Koala can share.
Thank you very much, Lisa. Thank you very much to the audience. So my name is Lassane Koala. I'm working at RSS here in Burkina Faso. I'm going to make a presentation on onchocerciasis vectors, the black flies. So the title of my presentation is Prospect for Developing Efficient Targets for the Genome Monitoring and Control of Similium damnosum, the major vector of onchocerciasis in Africa. So as a small introduction, and as the previous speaker said, the sticky target or insecticide target are efficient tools that are commonly used to catch or kill insects which are responsible of vector-borne disease such as malaria, trypanosomiasis, or onchocerciasis. These tools are cheaper, easy to deploy, and are very sustainable for our endemic country. So for the case of onchocerciasis, there was few studies who investigate the development of target, either for the genome monitoring or for the control, like for the CC flies. You can see at the right images some example of target that have been currently used for CC flies and also. So that makes the HLC remaining the main sampling method for the genome monitoring and all the strategy of vector control that we use against black flies are costly for our endemic country. So my global objective in this PVEC was to develop efficient targets that could be used to support the genome monitoring of the vector control in onchocerciasis elimination framework in Africa. So the development of such targets require a better understanding of Similium damnosum response to visual or olfactory cue released by the host. So the global objective of this presentation and of this result is to investigate the Similium damnosum response to different design of targets, which may make the human host a visual and olfactory cue in order to develop efficient device to support genome monitoring and vector control. So to do that, we conduct different activity in the southwest part of Burkina Faso, which is still endemic to onchocerciasis. We work in 3 black fly breeding sites, and we conduct 4 experimentation series. And in each series of experimentation, we assess the response of fly, for example, to a single stimulus or to a combination of different stimuli. So we conduct the comparison of different design of targets. You can see at the right images the different design of target that we evaluate. There is different size of target, different shape, and also different odors that we evaluate as well. For example, we assess the coca blend, vigilar, and also the carbon dioxide in combination. So to evaluate these different designs, we use the electrocution grids and also sticky target. And the main method experimentation design in the field or Latin square design. So through the experimentation, we measure different indices. For example, we calculate the number of black fly collected by each target per day. And in the case where we compare different size of target, we also evaluate the number of black fly collected or killed, in other words, per target area in meter square. We also measure the landing response of black flies on target. So at the end of all the experiment, we conducted 8 experiments, which allow us to identify the better size, the better shape, and the better combination of odor that could be used as a target to catch Similium damnosum black flies. This data has been published in Medical and Veterinary Entomology. I warmly recommend you to read it. So in terms of global result, we collect high diversity of flies, mainly host-seeking of Similium damnosum fly, but also repositing flies, new emergent, but also huge important number of Glossina, Stomoxis, rise up as well, and also tabanus flies. So in terms of xenomonitoring, with the global aim to collect the maximum number of flies to assess the infection, our result reveals that for a target, optimal design for a target to collect the maximum of flies should be stick a target of smaller size from 0.12 to 0.5 meters square with horizontal shape, and bite it with the Boca blend, which was the most attractant artificial bite that we used. You can see the final prototype that in the right images below. And with this prototype, we collect a very important number of flies relatively compared to, for example, a target bite and carbon dioxide. So we think that we could deploy several targets of this prototype to assure the xenomonitoring of onchocerciasis in Africa. And when we report the number of flies collected by the different target to the targets as a size to the target area, the result revealed that the smallest target from, for example, 0.25 meter side or 0.5 side also were the most efficient. This is important finding because so with a small material, with a little material, we can collect as the number of flies as a larger target. So for the vector control, we can use this target and protect them with insecticide and bite with Boca. Use it to collect, to catch and kill the Simulum damnosum in the vector control framework. The activity are still ongoing, and we complete some activity, and the result will be shared with you in due record. So in the next step, we want to deploy the target that we developed to capture black flies in onchocerciasis xenomonitoring. You can see in the images one of our activity in the breeding site. It was particular where we expose different design of target and which allow us to collect more than 2,000 of Simulum damnosum in 9 days. So we want to implement our target in large scale to see if we can use it for the xenomonitoring, and also the next step will be to use it to reduce the nuisance of black flies as vector control tools. So I'm at the end of my presentation. I would like to thank all of the persons and the institutions that support our work since the beginning of PVEC. Thank you very much.
Thanks very much, Koala. You can stop sharing your screen.
Okay.
And I'll invite Dr. Marcel Sandou to take the floor.
So as a background, malaria represents one of the most devastating diseases in Cameroon, more particularly in Africa. To fight against this disease, vector control has been one of the key elements based on the use of insecticide through impregnated bed net, indoor residual spraying, and larvicide. But these control measures are facing to some challenge. We can have insecticide resistance, limited numbers of insecticides, change in vector behavior, and the absence of effective malaria vaccine. Faced to this situation, it is very important to develop new alternative methods for controlling malaria transmission. And nowadays, one area of research involves mosquito-microbiota interactions that may lead to new tools for mosquitoes and mosquito-borne disease control. But the composition of microbiota is highly dynamic, vary greatly with species, stage of mosquito development, geographical localities, and season. For example, previous studies done in Ghana and in Mali show that seasonality and locality affect the diversity of microbiota in Anopheles gambiae and Anopheles covesi. However, there has been no work to date on the spatio-temporal variability in microbiota of Cameroonian mosquitoes. That's why this study aims to evaluate the composition and structure of microbiota in Anopheles covesi and Anopheles gambiae collected from 2 different localities during the dry and the wet season. So this study was conducted in 2 localities in Cameroon, namely Gunugu in the north region and Bankeng in the center region. Here, mosquitoes were collected using electrical aspirator during the dry and the wet season. After collection, mosquitoes were identified and some DNA was selected randomly for Illumina sequencing. Then the sequence data were analyzed through QAM and R software, and amplicon variant sequence were used to generate firstly the diversity indices, alpha and beta, according to the localities and the season, and secondly to taxonomy analysis at the different level. As a result, a total of around 2,000 mosquitoes were collected and identified as Anopheles gambiae. The distribution of the mosquito species between the 2 localities was similar, but in the rainy season, there were more mosquitoes compared to the dry season. It is also important to mention that Anopheles gambiae was collected only in Bankeng and Anopheles comisi was collected only in Gunugu. Among this collection, mosquitoes, around 120 mosquitoes have been selected randomly for illuminant sequences corresponding to 30 mosquitoes per season and per localities. Analysis of microbiota diversity was first described using alpha diversities with 2 indices. You have Shannon and observed amplicon variant sequence in order to estimate the inter-individual variation. Thus, for Anopheles gambiae collected only in Bankeng, no significant difference in observed and Shannon indices of microbiota were noted between dry and the wet season. While for Anopheles coryzae collected only in Gunugu, significant difference in observed and Shannon indices of microbiota were noted during the dry and the wet season. To compare the difference in microbiota composition and structure between seasons and Anopheles species from different localities, beta diversity data shown high distinct clustering by season. In pink, we have a dry season, and in blue, we have a wet season. Also, during the dry season, Anopheles gambiae and Anopheles coryzae had more similar bacterial community. While during the wet season, the 2 Anopheles species show a significant difference in bacterial composition. A special attention was paid to bacterial symbionts due to their role in the mosquito's interaction with Plasmodium. Here, a total of 4 major bacterial symbionts were detected, and their relative abundance was very different according to the season and localities. For example, during the dry season, Azaya is more predominant in Bankeng, and Pantoe is more predominant in Gunugu, while Serratia and Klebsiella are most predominant during the wet season in both localities. In the conclusion, we can say that this preliminary study provides valued indication that the season and were significantly affected both the bacterial composition and relative abundance of the bacterial genus. In addition, this study provides the evidence of the presence of potential bacterial symbiont that can be used to develop novel approach for mosquitoes control. Before to finish, I will seize this opportunity to thank Spivak for this wonderful 4 years that I learned a lot, notably in grant writing, data management, development skill, and so on. I will also thank all our collaborators from ICM and CREED. Thank you for your kind attention.
Thanks very much, Marcel. You can stop sharing your screen, and I'll invite Francine Sado to the floor.
Okay, thank you. I'm Francine Sado, PhD student from the University of Buea, and I conducted my research at the Center for Research in Infectious Diseases under the PIVEC project of Dr. Iget Simo. My presentation is entitled Infestation Rate, Seasonal Distribution, and Genetic Diversity of Isogenic Ticks from livestock of various origins in 2 markets of Yaoundé, Cameroon. As a background, chicks are obligate hematophagous that feed exclusively on the blood of their vertebrate host, and they are efficient vectors of pathogens such as viruses, parasites, and bacteria to humans and animals, and are the most important vectors of disease in domestic animals. Approximately 80% of the world cattle population are at risk of chicks and tick-borne diseases, and the economic losses due to tick control program and the disease they caused are estimated at up to 20 billion per year. There exists mainly for hard ticks 3 types of life cycles. We have 1 host tick, 2 host ticks, and 3 host ticks. And here I presented an example of exotic ticks with 3 host ticks. So the chicks has mainly 4 development stages, where we have the eggs and larvae, nymph, and adults. And on the adult stage, we can differentiate the male from the female. On figure 2, it shows the different parts of the chicks that were used for morphological identification, as described by different authors. In Cameroon, there is limited data on the seasonal distribution and the genetic diversity of chicks. That's why we aim to determine the infestation rate, seasonal distribution, and species diversity of chicks in livestock in Yaoundé's markets. To achieve these objectives, we conducted a cross-sectional and descriptive study in 2 markets of Yaoundé, mainly in the Fundi department, in the cattle market of Etudi and Tinga market, where we collected chicks on sheep and goats. In the cattle market, we collected chicks on cattle. Chicks were then transported using falcon tubes into the lab, where they were morphologically identified using described available keys. And they were then watched, and DNA was extracted. We then amplified the cytochrome C oxidase 1 and 16S rDNA. After sequencing, we analyzed our sequence using Mega, DNSP, and TCS. As first result, in total, we sampled chicks on 9,000 animals from Cameroon, Chad, and Sudan. And those animals was mainly composed of 6,000 cattle, 1,000 goats, and 900 sheep. And 2,000 was found infested by ticks. That makes an overall infestation rate of 28%. And with 2,000 from cattle, that is 94%, and 107 from sheep, 51 from goats. So, and 3,700 chicks were found, 3,731 adult, and 21 nymphs. 3 genera of chicks were described, and here we can find that the main one, the one that was made of much species, was Rhipisphalus species, followed by Ambioma species, and then Yaloma species. And all those chicks were composed of comprising 11 chick species. And the main species of chicks found were Rhipisphalus decoloratus and Rhipisphalus microplus. So for the infestation rate, we found that there was a high infestation rate in cattle compared to small ruminants, and high species diversity of chicks were found. 11 species was described. And for the seasonal distribution, we found that Rhipisphalus species were mainly predominant during the rainy season and Yaloma during the dry season. Concerning the genetic diversity of the chicks that we have described, we found that we use here to present the main cattle chicks that were found and the main chicks that were predominant in sheep and goats. For cattle, Rhipisphalus decoloratus was the main one, followed by Rhipisphalus microplus. But Rhipisphalus microplus was previously described in Cameroon, and it is a worldwide distributed chicks that is implicated in several that has been shown to be resistant to several acaricides. So it poses a real problem worldwide. And based on the 16S rDNA analysis, we found that 3 haplotypes were found from our sequences and 7 substitution sites. Concerning the maximum likelihood tree, 2 clades, our sequences form 2 different clades for Rhipisphalus microplus complex. And The two clades were close related to those from Cameroon and Malaysia. Concerning the COX-1, the cytochrome C oxidase 1 gene, we found that the sequence formed 5 different haplotypes with 56 substitution sites, and the ML3, the maximum likelihood tree, on the maximum likelihood tree, 4 clades were formed, and those clades cluster with sequences that was previously described in Cameroon and in Burkina Faso, and all those clades was found in clade A. So our Epicephalus microplate sequences analyzed cluster with the reference sequence of Africa into clade A that was already described by Silatsa and colleagues in 2019 in Cameroon. Concerning the genetic diversity of the main chicks found in sheep and goats, that chick was Epicephalus sanguinus, and it is known as the main vector of some zoonotic diseases such as Coxiella, zoonotic pathogens such as Coxiella burnetii, Escherichia, and Rickettsia. And based on the analysis of the 16S rGNA, we found that the haplotype network was made of 4 haplotypes with 16 substitution sites. And the maximum likelihood tree consisted mainly of 2 clades, and those clades cluster with sequences already described in Argentina, USA, Portugal, and also the clade 2 with Aruba, La Réunion, and Thailand. Concerning the cytochrome C oxidase 1 analysis, 8 haplotype was found and 56 substitution sites, and the maximum likelihood tree show the formation of 5 clades that cluster with sequences described in Brazil, Portugal, and China. So at the end of this analysis, we found a high haplotype diversity with the cytochrome C oxidase 1 sequence compared to the 16S rGNA. So I will not finish this presentation without thanking the institution that was involved in this project, the funders, and also all the team that was implicated in this work, particularly Dr. Riget Simo, that assisted me from the beginning till the end of this project. So thank you.
Thanks very much, Francine. So I'll invite Dr. Hyacinth Toei to the floor.
Hello to everyone. Do you hear me?
Yes, that sounds good. Go ahead.
Okay, thanks so much. Thanks for giving me the opportunity to talk about the topic, which is about the insecticide resistant Aedes from Burkina Faso and the evidence of the first evidence of the V410L bacterial mutation in Aedes from Burkina Faso. And then to start, you can say Aedes transmit a range of arbovirus bat infection, such as dengue, yellow fever, chikungunya, and Zika. You know that this arbovirus bat infection is really present in Africa, where more than 70% of the African population are at risk of one of these arbovirus bat infection. I think also back in Burkina Faso, as other West African countries, experimented many dengue outbreaks. In Burkina Faso, I think you experimented 2 dengue outbreaks. The first one was back in 2013, and this bigger one was in 2017, where you got about more than 15,000 dengue cases every year in Burkina Faso. You have more than 80,000 dengue cases occurring in Burkina Faso. I think really that is really underestimated. I think we are more than this one. And also, you know that, and then to react to the dengue outbreak, the most strategies are based on the insecticide use and odor spray. But the choice of the insecticide can based on the local evidence. Yeah, I think in Burkina Faso, as other African countries, there are very few data about the insecticide resistance overall. I think this nice paper, I think published recently, I think presenting the recent data on the study of the insecticide resistance in Aedes from Burkina Faso. And also this paper also presenting, and there's the data on the insecticide resistant mechanisms in the Aedes mosquitoes from Burkina Faso. How do you proceed? I will work on 2 cities, Bamfora and Ouagadougou, where you sample mosquitoes as larvae and eggs in urban and peri-urban site from July to November in 2019 and 20, 2 years time. And also we perform WHO tube bioassay based on the WHO procedure. And also we perform the intensity bioassay and the PBO assay to assess the implication of the monooscillaries in the insecticide, and also you test, and also for the mosquito longevity following exposure to the dexamethasone. And also to investigate in the implication of the target site mutation, we use the real-time multi-curve analysis for screening of the 2 main candidate mutation, the 1534C, the 1016P, and also the V410L mutation. What we have as a result here, you can see here on this graph, Aedes mosquito from Burkina Faso are resistant to paraffin and then to carbamide. And now in this paper, we report for the first time resistance here to organophosphate insecticide classes with moderate resistance to malathion and also to the pyrimidine methyl. And also we will use, and then the PBO assay here to test the implication of the cytochrome bat enzyme but involved in the resistance. As you can see here, pre-exposure to the PBO increased significantly the mortality, but did not restore the complex susceptibility. That means that as a resistant mechanism, the cytochrome is playing the role in the insecticide, but there are other insecticide resistant mechanisms, which probably the target site mutation. And also we did the intensity test to assess the level, the strength of the periphery, the level of the peripheral resistance. Here, I think you use 2 methods. We change first the insecticide concentration paper, and also we increase the exposure time. As you can see here, even if after 4 hours of the exposure to 0.3 tetrametry, the mortality was around 50% of mortality. That means that the Aedes mosquitoes from Burkina Faso are highly resistant to these insecticide classes. Interestingly, you observe the mosquitoes after exposure to 1 hour, 2 hour, 4 hour tetrametry. And then you notice that mosquitoes surviving 2 hours and 4 hour exposure are very short lifetime for about 50 days. That is quite interesting, even if they are really highly but interesting. I think they are a short lifetime. And also we screen for the 3 KDA mutation. The first one here was the F1534C, and then also the 1046X, which were previously reported in Burkina Faso. I think, and then the high frequency was obtained for this 1534C mutation from 0.35 to 0.92. I think when you compare the frequency between the 2 cities, in Ouagadougou, I think the frequency was really high than the Bamfora area. And then when you also to compare from urban to peri-urban side, high frequency was obtained in urban side than rural side. That's quite really interesting. And also we and then we access the implication of this KDA mutation in the insecticide resistance. And then you first expose mosquitoes to 4 hours, and then we try to screen the 3 mutations between dead and live. As you can see here, you observe high frequency of the 3 mutation in a live mosquitoes, but here the difference were only significant for the 1534C mutation. I think this paper is quite the first one to report the multiple insecticide resistance in Aedes mosquitoes from Burkina Faso. That is quite really a worrying situation, but also but also is the first time also we are reporting the V410L mutation. That means that Aedes mosquitoes from Burkina Faso are becoming really highly resistant, and then this is quite a worrying situation for the choice of the new insecticide which can apply in case of the dengue outbreak. To learn more about our work, I think you can go to the paper to learn more about the other data that presented in the paper. Thanks so much for your attention. Thanks to PVEC and all the partners and all the co-authors. Thanks so much for your attention.
Thank you, Toy.
Good morning, everybody. I am really pleased to present you my research paper entitled Lack of Robust Evidence for a Wolbachia Infection in Anopheles gambiae from Burkina Faso. You know that Wolbachia and metracellular bacteria is well known for its ability to manipulate host reproduction, enabling it to spread into insect population. Additionally, Wolbachia can inhibit the development of diverse pathogens, which makes Wolbachia an attractive agent for pathogen control. For example, the presence of Wolbachia in Aedes aegypti mosquitoes impair infection with dengue and other arbovirus. In Anopheles gambiae mosquitoes, some studies have reported a negative correlation between positive mosquitoes and Plasmodium development in natural population. But the use of Wolbachia in this species has been limited by challenges in establishing stable trans-infected lines and uncertainty around native infection. High frequencies of infection of Wolbachia have been previously reported in Anopheles gambiae, collected from the Valley du Cou region of Burkina Faso. Here, we evaluated the occurrence of Wolbachia in natural samples collected from this region, 12-year time span, and in addition, expand sampling to other sites in Burkina Faso. Mosquitoes were collected in 11 villages during the rainy season. These sites are located in the Sahelian zone and Sudan Savannah zone of Burkina Faso. And more than 5,000 adult Anopheles gambiae mosquitoes morphologically identified the 16S urina gene. All samples were also analyzed by nested PCR. Then, all positive amplicons were sequenced, and finally, phylogenetic analysis of Wolbachia samples were performed on the conserved 16N urina sequences. In this study, no amplicon positive samples were found with the regular PCR. In terms of the prevalence with nested PCR, 46 amplicons were positive for Wolbachia, representing a prevalence of 0.93%. The nested PCR was repeated for each of the initial 46 positive mosquitoes, and 29 remain positive on the repeated PCR, representing a prevalence of 0.59%. The inability to confirm 17 individuals in a repeated nested PCR suggested the template is at the limit of detection in these samples. Regarding the spatial distribution, all positive specimens were found in just 3 of the 11 villages. Phylogenetic analysis of the 16S ELENA gene sequences used in these studies resulted in a corrected alignment of 317 nucleotides in length. A midpoint rooted tree showed that all sequences obtained from the study clustered with supergroup B, Wolbachia. Contrasting with the sequences from the study, previously published sequences from Anopheles gambiae were observed to be distributed throughout both supergroup A and supergroup B. Our data indicate that Anopheles gambiae are free from Wolbachia, or if these are true infection, the low prevalence in the population suggests it is challenging for the bacterium to establish within the population. While some of our applicants were single sequenced and had similarity to Wolbachia, we cannot discount the possibility these were false positive. Regardless, the lack of preeminent native infection in Anopheles gambiae is encouraging for application utilizing Wolbachia trans-infected mosquitoes for malaria control. Here is our research team. Thanks for your kind attention.
And I'd like to invite our final speaker, Dr. Justin Pufford.
Thank you, Lisa. Okay, and before I start, I should make a few points about this presentation. First, it's going to be quite different to the ones that have come before it, in that I'm not presenting the results of a piece of vector control research. Rather, I think, as you all know, the PIVIC program at its heart was a capacity building program designed to build research and leadership capacity in a cohort of young African scientists. So that this presentation will focus on those factors that influence the effectiveness of the capacity strengthening program itself. So what we learned through the experience of implementing the PIVIC fellowship scheme. I also wanted to note, this is not one of the publications in the special edition, so you won't find it there, but we are writing this up for publication. It has yet to be submitted, but it will be shortly. And finally, although I'm Justin and I'm presenting this, the work was led by Jessica Amaji-Quach, who was unable to present today. So this is really a presentation on her behalf. So the study objectives, I think the primary objective was to understand those program factors that enhanced or hindered the capacity development of PIVIC's African postdoctoral research fellows. In terms of the study design, it was a longitudinal qualitative study that spanned the full 4 years of the PIVIC program. Data were primarily collected by focus group discussion and in-depth interview conducted with the postdocs themselves, but also PIVIC leadership, the supervisors and advisors for the postdoc fellows, and various research support staff. This slide just briefly presents the participant characteristics. So across the 4 years, we interviewed a total of 28 people, but I should point out many of these people were interviewed more than once. So in terms of the number of transcripts we have to inform our data analysis, it's a lot larger than 28. We also drew on a lot of PIVIC documentation to inform the analysis. But in terms of the overall 28 participants here, you can see they came from both, the southern and northern partner institutions, from many different positions within the consortium, as previously mentioned, from across all countries involved, and indeed language groupings. So I'm not going to spend too much time on methodology. I'm just going to jump straight into two sort of key results slides. The first here, this slide depicts program factors that were considered more or less supportive of early career researcher development or capacity strengthening. So supportive factors in green, less supportive factors in red. I'm not going to read through each of the bullet points. I think you'll see that on your screen, and please feel free to do so. I'm just going to highlight a few from both the supportive and less supportive side. So starting with supportive, I guess certainly what we found within PIVIC and indeed similar programs elsewhere, providing a wide range of networking opportunities is really important for early career researcher development. So this is, opportunities to network with other researchers from your own and other disciplines, networking with key stakeholders, research end users, community groups, and so on and so forth. So the point here is that networking opportunities are valuable, and particularly diverse types of networking opportunities. I think another factor that worked well within the PIVIC program was providing the research fellows with quite a lot of research autonomy, or well-supported research autonomy. And in addition to that, multiple responsibilities beyond just the design and implementation of this study. So this might include such things as supervising masters or PhD students, actively participating in management board meetings and these sorts of things. And another point I'd like to highlight was simply the cohort approach. The fact that there were a large group of fellows working through this program together allowed strong peer networks to develop and peer support to take place, which also seemed to be a really supportive factor. On the less supportive side, a number of the postdocs were recruited from existing positions within the partner institutions, which meant that although the fellowships were meant to be 100% full-time, they often retained some institutional responsibilities that sort of predated their entry to the fellowship. And on occasions, this could create some tensions and additional burden in addition to what was quite a demanding fellowship process. All fellows were provided with advisors and mentors, and generally this worked really well. In some cases, the matches were perhaps not as ideal as they could have been, or were not as productive as they could have been, and perhaps some scope to change mentors or advisors might have been useful here. And the third point to highlight, although PIVIC was a research-driven partnership and produced, as you've seen, a lot of really wonderful research outputs, it also had the strong capacity strengthening focus. So fellows not only undertook their research, they engaged in a lot of training activities, a lot of networking activities, a lot of other activities to develop well-rounded researchers. And I think this point is recognizes that when you do that, additional time is needed. It takes a lot of time just to implement a research project, but when you add all these additional elements to it, it can become quite overwhelming for the fellows, and more time may need to be factored in for that. So all of these factors, I think they reflect the common issues from across all interviewees, and in a way, it's just reflecting their voice back to you. But we did take the analysis one step further and tried to identify overarching factors like key themes, key lessons that came about from the PIVIC experience. And that's what I want to touch on in this last slide. So I think for any listeners who are interested in putting together their own partnerships and developing their own capacity strengthening programs, these might be particularly interesting. So firstly, I think what we sort of came to understand is that the fellows' recognition of the broad skill set needed for scientific career advancement, and indeed scientific career leadership, was certainly aided by their exposure to a diverse work and training mix. So, understandably, as a young scientist, when you start a research program, your mind can be really trained on the design and implementation of the study, and all of the additional things that are important in a research career are perhaps not prioritized. but by being exposed to those additional things, their importance becomes apparent and the appreciation for them becomes apparent. So that exposure, again, is a really important element of a capacity strengthening program. The second point, or second key lesson, is recognizing and managing competing interests and ensuring all partners benefit from participation. So again, why PIVAC prioritized the research fellows as in terms of investment and focus of the program. There were a lot of other groups also involved in both supporting the fellows and in doing their own things as well. And so ensuring that the partner institutions also benefit from participation, that the fellows advisors and supervisors also benefit, that there's a tangible gain for everybody involved is quite important. The third point or third key lesson was around flexibility, so that the PIVAC program had a lot of sort of predetermined elements that were rolled out over the course of the 4 years in terms of the amount of money available for each student or each fellow, to undertake their research, in terms of training opportunities and the like. And this fixed content was really valuable, but each fellow is also an individual and has individual needs, and having flexibility to adjust some of those fixed provisions may also have enhanced capacity strengthening. Fourth point is around ensuring the equity of opportunity and rewarding engagement. And that when we look at the outcomes across all of the fellows and everybody who participated in PIVAC, perhaps not surprisingly, those who were able to engage the most perhaps benefited the most. And whilst on paper and principle, everyone should have had the same opportunity to engage fully, this was not always the case. So, for example, when you have pre-existing responsibilities in the workplace, perhaps caring or personal commitments in your private life that can prevent full participation. Recognizing that people have different restrictions in their life, and again, some flexibility to ensure everyone does have equitable opportunity to engage is quite important. And once you can be satisfied that there is opportunity for equitable engagement, then those who do continue to engage at a higher level perhaps could be rewarded for that. So really enhance and make the most of their learning opportunity. And finally, this entire presentation, if you like, is a result of reflective practice. As PIVAC was rolled out, we wanted to understand what worked well and what worked less well, and engaging all participants and informing that out in that process, which I think was really valuable and generally worked quite well. Although it also became apparent that perhaps not all groups within PIVAC were equally comfortable providing constructive feedback. So providing recognizing that there might need to be different strategies used and different approaches to ensure constructive feedback from everyone is perhaps quite important going forward. So I think that must be about my 10 minutes. So I'm going to stop there. So thank you all for listening.
Thanks very much, Justin. I'd like to invite all the speakers to turn on their videos for the last session, which is just a Q&A. And while you're turning on your videos, I just want to thank all of the speakers. It's been excellent to see the breadth and the diversity of research topics that were covered within this consortium. And you could really see how the research was really designed in response to emergent public health needs. It was great to have the scene set on the historical context provided by Moses, and great to hear from Justin and putting some of that reflective practice into action. So I'll hand over to Professor Hilary Ranson to chair the Q&A.
Thank you, and I'd just first like to echo my thanks to all the speakers. Really, really great to see all the progress made, and as Lisa said, the diversity in the topics covered. And we've had quite an active chat, and a number of questions have already been submitted, and quite a few of them have been addressed by the speakers. So thank you for keeping that active engagement throughout, and I would encourage all the speakers to keep an eye on that, and if there's some very technical questions there, it might be easier to address on the chat. But I'm going to pick out a few initially, both from the chat and a few that have come in by email. And I think I will start off with a question for Koala. And Koala, you talked about the large numbers of black flies that you collected. I think you said 2,000 in 9 days. And there was questions about comparing the trap method compared to human landing catches. And I think you've addressed that and said that you are looking at both methods. But I wondered if you can comment on whether the numbers that you are collecting with the current versions of the traps are likely to be sufficient to act as a control tool, or do you see these traps more as for surveillance? Koala, are you there? And can you hear me?
Yes. Can you hear me?
Great.
Can you hear me?
Yes, yes.
Sorry, it was broken. Could you repeat the end of your question, please?
So the question is really do you see the application of your trap for surveillance or for control, or both?
Okay. My connection is quite bad, but I think I understand the question is if I think the current design of target will be deployed as a monitoring tool. Is that correct? Or?
Yes, the numbers you would need to collect for it to be to have an impact on controlling the disease. Will your trap be used in that way, or will it be used as a monitoring tool?
Yes. Okay, I see now. We collected the 2,000 flies I'm talking about was collected in one experiment in Bakula. And for the monitoring, we need to collect around 6,000 black flies transmission zone. So with the current version, we can collect enough flies to conduct the monitoring in one site. It's depending on the times.
Mm-hmm.
This was we collected 3,000 flies with 4 targets at least, and in 9 days. So we think we can deploy it for because generally to assess the transmission is during the period of high transmission, which is generally in the rainy season. So we can deploy the target at this time and to collect the sufficient number of flies from the monitoring.
Thank you. Thank you.
Get me my connection?
Yeah.
Okay.
A question now for Moses. Moses, you referred to the need for equity-driven funding, and you cited a recent paper on this topic. And I wonder, and you have been involved with the PVET consortium from the start as the chair of our external advisor. And I wonder if, external advisory board, I wonder if you could comment on whether, given that the PVET consortium, the funding for PVET came to a Northern organisation, came to Liverpool School, whilst it had partners, many partners that we've heard from today, whether this sort of funding mechanism is perpetuating the inequities in we see in global health research, and what are the solutions to that, and what how do we flip this situation to ensure that research in vector-borne diseases is led by the global south?
Thank you very much, Hilary. I think with the establishment of the Africa Center for Disease Control, I think we are now getting enough engagement from African governments to enable that are contributing substantial amount of money now to research, especially with a lot of work here around genomics. So recently, the African Development Bank, I think, committed about $100 million towards that. So I think we are now, since the advent of COVID, governments have realized that, and the signs are good. So as soon as we get that established, I think the voice from the vector community will begin to talk about how this can move now. But the signs are encouraging.
Good. I'm very pleased to hear that. Thank you, Moses. Please do continue to put any questions in the chat that haven't been answered already. But I will turn to a question now for Toei. Toei, you presented data on resistance to different classes of insecticides in the Aedes mosquitoes in Burkina Faso, and really showing a picture of multiple cross-resistance to different classes. What are the implications of this, and what's the messaging to the Ministry of Health and the dengue control program in Burkina Faso in light of this multiple resistance?
Okay, thanks so much, Larry, for your question. I think in the paper is the first time that you are presenting multiple insecticide resistance in Aedes from Burkina Faso. I think first conclusion, as you can say, that as in Anopheles gambiae in Burkina Faso, where multiple resistance is observed, and also in the Aedes mosquitoes, we are also seeing the same pattern you have already in the Anopheles gambiae one. I think the message here, first, we need to continue the monitoring of the insecticide resistance in the Aedes mosquitoes. I think because we work only in 2 cities, yeah, I think you can extend the monitoring. That could be implemented in the national strategy, and then to continue the monitoring of the insecticide resistance in Aedes mosquitoes, as we are already doing for the Anopheles mosquitoes. And also you can extend this work to the other new insecticides which can be used, as for larvicide, for instance. Yeah, that can be the alternative. I think you can also continue looking for the new insecticides which can be used in case of the dengue outbreak in Burkina Faso.
Thank you. And just are any non-insecticidal methods being considered for dengue control in Burkina?
Yeah, I think in Burkina Faso we are trying to work with the population about the water holding cotton, then cleaning. That can be also the alternative, yeah. But that and I think for that we just need to train the people to show them where larvae then can be found, and then for cleaning in case of the dengue emergence and dengue outbreak back in Burkina Faso. That can be the no insecticides but solution.
Thank you, Terry. Important work. So we've got a question about which I guess I'm going to put to Justin, about has your analysis on the capacity strengthening work thrown up any insights into gender-based differences in both in the recruitment or the advancement of male versus female early career researchers in the program?
Thank you, Hilary. I would say, I mean, the sample we're working with is quite small, so I wouldn't necessarily say that is immediately apparent. Although there are, if we look at the list of supportive factors for the PIVOT program, for example, one thing that really stood out was in the initial selection process. This was a competitive program. All potential fellows had to apply and be, screened and go through a selection process. And I think we got really positive feedback from both, male and female fellows, that the fact that African partners were highly visible in that selection process, that interviews took place physically in African institutions, and where necessary, French language, for example, could be spoken or accommodated in that selection process. This made it a far more accessible opportunity for many fellows as compared to their previous experiences applying to other international programs. So I think in terms of improving the access to these types of opportunities for both males and females, it was a really important and valued feature of the PIVIC program.
Thank you. Yeah. Thank you. And yes, do accept that. Yeah, too small a sample size to draw any real conclusions there on. But yeah, thanks for your comments. We are drawing to the end of our time slot. There are quite a number of questions that have been posted in the chat, a lot relating sort of to seasonal variations in abundance or species composition, etc., of the various different vectors that we've heard described today. But I think most of those have been addressed by the speakers. So thank you very much both to those posing the questions and to our speakers for answering so promptly. And hopefully this has made some new connections, and there'll be some follow-up discussions on these topics after this session has finished. But I'm not seeing anything further coming through at the moment, so I think I'm going to just pass back to Lisa. Well, I'm going to thank, I would like to just thank Medical and Veterinary Entomology, the editors and all of the journal for this special issue and for all the work in putting this webinar together. And again, just encourage everybody to check out the papers that are now online. And I'll pass back to Lisa to close the session.
Okay, thanks very much, Hilary. And I kind of wanted to echo the same and thank the Royal Entomological Society for hosting this webinar. Moses for all the hard work behind the scenes in editing the special issue. So please do go online. It's all open access. Check it out and share it.